Here's what they did. They compared the 40 known hobbit teeth with those from 490 modern humans from Asia, Oceania, Africa, and Europe, as well as from a variety of extinct hominins, such as Homo habilis. And some of the hobbits' teeth were small, as small as those from, say, short modern humans. But other features in other parts of the mouth, or the teeth, I should say, looked completely dissimilar from those of modern humans. Here's what they told us specifically. The canine and premolar teeth looked primitive, whereas the molar teeth looked advanced, as if they had emerged later in the evolution of Homo sapiens. That's what the scientists said. So their findings contradict earlier claims that hobbits possess teeth entirely like those of modern humans. So another piece of the puzzle has been entered into the arena, and now it's for the professionals to go back and forth and figure out if this will sway anybody into thinking differently about their prior thoughts.
Yeah, I've been following this from the beginning. It's like every time one side or the other published a study, it's like, we're right, no, we're right, no, we're right. It just keeps going back and forth depending on how you look at the evidence. The problem is that I suspect that this debate will not be settled with the existing fossils.
And
so
we'd have to find
too small a sample size.
So if we find new fossils of clear-cut Homo floresiensis, well preserved with, some cranial and extracranial bones, that will settle the debate. If we never do, the longer we fail to find further specimens, I think the less robust the argument will be that it's a new species. But it will never really fully settle the debate. I think there'll still be holdouts. So it'll be a lot easier to prove that it is a new species than that it isn't a new species, I think. But I don't think this is going to be settled until new fossils are found.
If that's the case, Steve, what's lacking in the fossils that we have at this point?
Just number of specimens. Because you could still argue that one skull we have is a diseased human.
Right. We've got the one skull, and we've got several fragments of skulls, like several jaws.
That's it. If you have one specimen, it could be anything. If you have two, it's got to be a species, right? So that's the issue. We need a completely separate, complete enough specimen to say, yeah, this is absolutely a new species. That will end the debate. Until then, they're going to, I think, dicker over the minutia. It's one time it's the cranial capacity or the shape of the cranium, and now it's the teeth, and then it's the facial bone structure. So it's
the wrist bone. They were even looking at the wrist bones or the arm bones of the critter and saying that's human.
Why are you calling it a critter?
Because it's a hobbitsis.
It's easier to say.
It's still a person. It's just not maybe a modern human.
I call people critters.
Okay, all right. I love that the writer of this article, Charles Choi, who I have to admit I know personally, and I think he's a great science writer, but I love that his headline called the teeth chompers. Because anytime you can say chompers instead of teeth, it makes me really happy.
Or critters instead of people.
Yeah,
or
okay.
Critters
with chompers.
Chompers of a critter. All right. Well, Kara, you're going to tell us about mosquitoes and malaria.
Kind of the headline that we're discussing here is a new type of anti-malarial mosquito. And in order to understand this mosquito, you have to understand the technology behind it. So the technology is called gene drive technology. And like I said, it could mean the end of malaria as we know it, but it's probably going to have to pass through a bunch of technical and ethical hurdles first. So in order to understand this, we need to understand what a gene drive is. It's still pretty controversial. It's been on researchers' minds for over a decade. They've been theorizing it, and they've been working on how to make it work. But earlier this year, there was a study that showed that it's highly, highly effective. So basically, in essence, the gene drive is a tool that's used to bias inheritance in offspring, causing a larger percentage of the progeny of a certain species to carry the desirable trait that you're looking for. So it's, think of it this way. It's an engineered piece of DNA that copies and pastes itself into the genome. And then when the two organisms mate, the gene drive inherited from one parent actually inserts itself into the gene inherited from the other parent, so that the offspring have a significantly higher than 50% chance of having that trait, whereas in normal inheritance we see like 50% 50% on average, depending on the type of inheritance that we're talking about. It's really useful now. Like, it's become more advanced because of this CRISPR technology, which you've probably heard of. CRISPR. It's actually an acronym that stands for Clustered Regularly Interspaced Short Palindromic Repeats. But CRISPR is basically a genetic modification tool using Cas9, which is a specific protein. In this case, they use Cas9, and that helps researchers delete, add, activate, and even deactivate certain genes. The reason that it's such an improvement is because it happens fast, it's pretty accurate, and it's way less expensive than it used to be. And it uses plasmids, which is, like bacteriophages, like viral bacteria, which is something that researchers have been doing for a while. This is just a huge improvement.
Yeah, the system, by the way, was derived from bacteria. It is a method that bacteria use to fine-tune their immune system against specific viruses.
Yeah, that they'll over a series of viral, punishments, they will make their immune system more specifically geared towards that. And they were able to use that natural process in bacteria and then basically harness it in order to splice these genes and to put in genes that they want to do certain things. So let's talk about what those genes are. Before we talk about what those genes are, let's talk about the example from earlier this year, which was wildly successful. There were researchers at UCSD who used this technology, this gene drive technology, in a batch of fruit flies. And they expected it to sort of work, and it would take generation after generation in order to finally get a population that had the trait that they were looking for. They got it within like 2 generations, and it was something crazy, like almost 100% of the progeny contained the gene of interest. And they were like, holy crap, this works really well, and that is good, but it could be bad, and we'll talk about why. In the meantime, there was a researcher at UC Irvine, so another California institution, named Anthony James, and he'd been plugging away at this problem of malaria in mosquitoes, specifically a type of mosquito called Anopheles tefensi, and that's the Indian mosquito. That's the primary malaria vector in that part of the world. And he's plugging away at trying to figure out what to do about malaria in these mosquitoes. And he's already been successful at genetically engineering a mosquito to make antibodies against malaria using mouse genes. So he's done all this great work in the lab. And he's like, okay, I have this anti-malarial mosquito. How do I get it to spread? Meanwhile, this group at UCSD is like, holy crap, this gene drive is working really well in our fruit flies. So they get together and they say, what if my cargo, these genes in the mosquito that are anti-malarial, are carried by your gene drive technology? Maybe we can make a mosquito that when it breeds makes all of its progeny also have this anti-malarial property. But there were a lot of, problems, right, when they first combined forces. One of them was that the cargo was massive. We're talking 17,000 DNA bases carrying these necessary genes, but eventually they figured it out. Also, when they first tried, the gene drive took in only 2 male mosquitoes of the total 25,712 in the experiment. So they had over 25,000 mosquitoes. They inserted these gene drives into them, and only 2 of them took it. And they knew that because they also inserted a gene that made their eyes change color, so they could see them pretty easily under fluorescent light. The crazy thing is, even though only 2 male mosquitoes successfully got the gene inserted, the gene drive worked so well that by the 3rd generation, 98.9% of the male progeny of those 2 male mosquitoes carried the drive. Now, I know what you're thinking, because I keep talking about male mosquitoes. Isn't it the female mosquitoes that bite? Aren't they the ones that require blood meal? Aren't they the ones that spread? Yeah, and that spread malaria. Yes, that is true. And the researchers do admit that they've had a harder time getting the females to accept the drive for various reasons. It seems like once they insert into the DNA, it actually just closes in on itself, and it partially works. It splices the DNA, but they can't get the new package to insert appropriately. Also, the Cas9 protein that they're using in this CRISPR tool seems to be a little bit toxic to mosquitoes, so they have to be careful in their dosage and finding just the right delivery site. But the researchers are pretty sure that they can get past those hurdles and that this is a huge step forward. They agree that the gene drive won't fully eradicate malaria, but it could eventually get the parasite under control so that it's no longer a threat to health and human safety. The problem here